All right, guys. So today we're going to start doing chapter 10, which is on DNA, right? So the structure and the function of DNA. So amazingly enough, this wonderful structure of DNA that we see pretty much everywhere on social media and in all sorts of like symbols, etc., we're very familiar with it, right? It actually took a really long time to figure out this structure. And if you guys know things about biology, which you do by now, biology, how it works is for you to understand how something works and what the role that particular thing like DNA has in the world, you need to understand the structure of it. So let's look at how we're basically going to do a brief history like lesson on the discovery of DNA. So they knew it was a chemical component, like they knew it was found in cells in the 1800s, but they didn't know what it was like in the 1800s back then. They didn't really realize what the DNA was for, including really important people like Mendel. Remember Gregor Mendel, the father of genetics? He didn't know that DNA was the actual thing that carried his heritable units, or the thing that carried his genes and alleles, right? They had no idea what role DNA played. So in the 1920s, they identified chromosomes. So they saw that DNA packaged up into specific things known as chromosomes. But again, they didn't quite realize where the gene were. They didn't understand that genes were found on the DNA, right? They thought that they could see, you know, that they could see them under the microscope or anything like that, and it just didn't work that way. So they just, in the 1920s, all they realized was that chromosomes were actually made out of DNA. That's all they realized. And then in the 1930s, so not too long ago, about 100 years ago, that's when things started clearing up. That's when people realized that the molecule that actually carried the inheritance pattern from one generation to another was actually DNA. For a while, people thought it was the proteins that did it, but we know that proteins don't do that. And so in the 1930s, those things cleared up and they realized that it was indeed DNA. What was not understood, however, was how DNA did this, right? They didn't understand how DNA was passed on from one member of the family to the to the offspring. And in order for them to understand how DNA did this, they had to figure out the three-dimensional structure. So in come, these two guys. Watson and Crick. So James Watson, this guy on the left, and Francis Crick, this guy on the right. James Watson was a really young American, kind of like a hippie, I think you'd call it. Like, actually, I don't know if he'd call himself a hippie, but he would just like wear Converse shoes, and he was just very laid back. Francis Crick had just come from the war, right? He just finished up World War II, and they were both ready to basically start their careers in biology. No one knew who the heck these people were. They were look how young James Watson was. They were very young, inexperienced. No one knew who they were. And so at this time, since everyone found out that DNA, the molecule DNA, was actually responsible for for holding genes, and everyone wanted to figure out what the DNA looked like, even though today we know what it looks like. We know that it looks like this, right? Back then, they didn't know that it looked like that. Everyone wanted to figure out the structure. So it was this humongous, it's actually my favorite thing, this humongous race to figure, in the science world, to figure out the structure of DNA. Everyone was basically putting their efforts into finding this mysterious structure that makes up DNA. And really big named scientists as well of those times, like Linus Pauling, etc. But the people who actually discovered DNA, the structure of the DNA, were actually two little guys that no one knew about, James Watson and Francis Crick. And for their discovery, they then went ahead and actually won the Nobel Peace Prize, not the Nobel Peace, the Nobel Prize for it in 1962. Okay, so it was important for them to figure out the structure because the structure revealed to them how the information was passed on, and in addition, how DNA could be so versatile to make millions of proteins and to make so much genetic diversity. They couldn't figure out how DNA could allow for so much genetic diversity. So with their discovery, they opened up doors to a new field of biology known as molecular biology. This is just the investigation of life at the molecular level, right? Because they're looking at the molecular level of DNA. So how did they actually discover the structure of DNA? And we'll actually, even as they discover it, we'll talk about what the structure of DNA actually looks like. So the microscopes at that time were not powerful enough for them to see the really, really teeny tiny DNA. Instead, they used something known as X-ray diffraction, which is basically they would take a molecule of DNA, which is what I have right here. You guys know that it's double-stranded and it's helicized and wrapped around itself. And they bombarded it with X-ray beams, okay? These X-ray beams would hit the DNA and then scatter. And this scatter pattern, like when they scattered, they were collected by a specific X-ray film, right? And this particular scatter pattern showed kind of what the DNA looked like. And in fact, this is what came about, this particular picture. And you may look at this particular picture and say, like, that does not look like anything like DNA. And, you know, you kind of are correct. It doesn't look like the typical DNA that we look at. However, that's what DNA looks like when you see it from, like, so if this is my DNA, right? When you look at it from above, that's what it looks like. So they were able to start from that picture, which to us doesn't make any sense. At least to me, it makes no sense. From that picture, with the help of this other lady known as Rosalind Franklin, who was an expert at this technique, like the X-ray diffraction technique, they were able to figure out what DNA looked like. Unfortunately, Rosalind Franklin, since she worked so much with X-rays, actually died of cancer really early on. And so by the time Francis and James were awarded the Nobel Prize, she was dead, and the Nobel Prize does not get awarded to people that are already dead. So using wire models, Watson and Crick, and with the help of Rosalind Franklin, began trying to construct a double helix that conformed to the data that was given to them from the DNA, right? So at the end, they determined that the DNA is a double helix, and that, so it looks like this. So it has one strand of DNA here, another strand of DNA here, then it wraps around itself, right? So these two strands are connected to each other, and we'll see how they're connected later on. It wraps around itself and it forms this helix, so it twists. And so that's basically what they were saying, that the DNA is a double helix. And they also went into the actual structure of the component, like the subunits or the building blocks of DNA, and we'll get to that in just a second. But basically, in the end, they published a paper. It was actually fantastic. It was such a huge scientific discovery, and they published a 2-page paper, front and back, 2-page paper. That's it, on the structure of DNA. When someone usually would, you know, type up like a 100-page paper on the structure of DNA, they typed it up in 2 pages and they submitted it. And that's what was immediately accepted by various journals. And that was what, you know, people finally understood what the structure of the DNA looked like. So, and it was really important to understand the structure of the DNA, because remember, by understanding the structure, you understand how it completes its role. And so, in other words, the structure of the DNA points towards a molecular explanation for life's unique properties of reproduction and inheritance. So it explains how DNA reproduces and replicates itself, and how then it's then, like, those two things that it's just replicated are inherited in the successive generation. So the arrangement of parts within DNA affects its action within a cell, an example of the relationship of structure and to function. So DNA basically looks like a ladder, right? A ladder, one strand is one side of the ladder, the other strand is the other side of the ladder, and they're connected by the rungs. Each rung represents a pair of bases, and we'll talk about them in just a second. And they're connected, remember, they're connected. How are they connected? By hydrogen bonds. Yeah, we'll actually talk about that in a little bit. So, okay. So the components of a DNA molecule, okay, or any nucleic acid actually, so including RNA molecules, are known as nucleotides. And we mentioned this a little bit in Unit 1, so hopefully this isn't too new to you guys. But the components of these RNA and DNA molecules, which are basically all like nucleic acids, are known as nucleotides. So when you have more than one nucleotide connected to another, you get this polynucleotide, right? Many nucleotides. So there's a variety of different kinds of nucleotides. We have in DNA, we have 4 different types of nucleotides. And then in RNA, we have an extra one that's a little bit different. And these nucleotides look kind of funky. Okay, they have a specific structure to them. But before we go into what nucleotides, like what the DNA nucleotides look like, let's talk a little bit about the difference between RNA and DNA. Okay, so DNA stands for deoxyribonucleic acid. And we know what the primary role of this DNA is. The primary role of this DNA is to basically bear our information, to hold information, our inheritance information, that we then get to pass on this information to other, to our offspring. Okay, so this is basically our code in which we make all of that codes for everything that makes us who we are. Okay, so all the proteins that arrange and arrange to make us the human that we are coded by DNA. So DNA contains the instructions to build proteins. Okay, but it doesn't actually get involved with the actual building. It just is the code. All right, so it's kind of like a computer code. You know that every single program on your computer has to be put in or made through a code, right? So this code is going to make that program. Same thing with DNA. The code of organisms is going to make that specific organism. Change the code, you change the program. Change the code in an organism, change the DNA in an organism, you change the organism a little bit. So this code is made out of 2 chains, okay? 2 chains that are composed of, again, those subunits known as nucleotides. So it's 2 chains, that means it's double-stranded. Each strand is just a bunch of nucleotides. And then they're linked together. And then in addition to being linked together, they are twisted to form a double helix, okay? So again, it encodes a genetic instruction that we need in growth, development, functioning, and reproduction of all known living organisms, and even some viruses. Yeah. Then comes RNA. RNA is another kind of nucleic acid, but it now stands for ribonucleic acid. RNA is not a code. Actually, what we use RNA for is it's imperative, it's very important for how we actually read and make DNA into a protein. DNA, before it's made into a protein, has to be made first into RNA, and then from there, the RNA can be made into a protein. DNA cannot directly be made into a protein. There's always a little step in between. That little step is basically carried out by RNA. So RNA is essential to actually help make the proteins. If we didn't have RNA in our system, we would not be able to make proteins, even if we had the DNA or the code that encoded the proteins. RNA is just something that is needed for the synthesis and the creation of proteins. So it has a few different roles, and all these roles are essential for the synthesis of proteins. So we'll look at it in upcoming slides, but there's going to be something known as rRNA, also known as ribosomal RNA, or tRNA, transfer RNA, or mRNA, messenger RNA. All of these RNAs are necessary components for protein synthesis. Okay, so they can help read the DNA, the code that's found on the DNA, and make it, translate it into an actual protein. The other thing about RNA is that it's also made out of nucleotides. Now, however, the nucleotide is just 1 chain instead of 2 chains, okay? And then there's a couple of other differences, like some specific nucleotides are missing and others are included. And so we'll talk about the structure of DNA first. Now that we know the roles and what they do and why they're important, we'll talk about the structure of the DNA first, the structure that was discovered by James Watson and Francis Crick. And then we'll talk about the structure of RNA, okay? So the structure of DNA. So like I said, DNA is made out of nucleotides. There are 4 types of nucleotides that make up that make up DNA. These are known as adenine, thymine, cytosine, and guanine, okay? You actually don't need to know that this is double. Okay, adenine, thymine, cytosine, and guanine. Those are the names of the specific nucleotides. Okay, so if the nucleotides were yellow, not yellow, what the heck? If the nucleotides were Lego pieces, okay, adenine would be all the red pieces, thymine would be all the green pieces, Cytosine would be all the yellow pieces, and guanine would be all the white pieces. Does that make sense? So they're all the same thing. They're all nucleotides, but each are a little bit different so that they can be their own particular thing. So what is a nucleotide? A nucleotide actually looks like this, okay? A nucleotide has a phosphate group, which also is this right here, phosphate group, which is just a P phosphorus with a bunch of oxygens. It's then connected, this phosphate group is connected to a sugar molecule, okay? This sugar molecule, it's really important to remember, the sugar that makes up DNA is known as deoxyribose, okay? Hence the name, remember the name of DNA is deoxyribonucleic acid. It's called deoxyribo because the sugar in it is deoxyribose. Okay, so hopefully that shouldn't be too difficult for you to remember. All right, so deoxyribose is the sugar. And then in addition, they have what's called a nitrogenous base. So a nucleotide is made out of 3 different things, okay? 3 different components, 3. Phosphate, sugar, and then a nitrogenous base. This nitrogenous base can tell the difference, is going to be what's going to dictate which kind of nucleotide we have. So here we have cytosine in it, so this entire nucleotide is a cytosine. If we have thymine, the entire nucleotide is thymine. If we have guanine, the entire nucleotide is guanine. If we have adenine, the entire nucleotide is adenine. So this is basically what each nucleotide looks like. And remember, one strand of the DNA is just many of these nucleotides bound to each other. And so you see here, okay, you have one nucleotide. The blue is the nitrogenous base. The red is the sugar, specifically deoxyribose, okay? And then the phosphate group. This is one nucleotide. And then it's bound, at the bottom, it's bound to another nucleotide. Base, nitrogenous base, sugar, phosphate. And it's bound, that one in itself is bound to another one. Base, sugar, phosphate, etc. Okay, so now I'm going to attempt to draw the DNA for you guys. Okay, so basically we have one strand of DNA, and I'm going to draw one strand first, and then another strand of DNA. And remember, these 2 strands are connected to each other, okay? We want to form a ladder. All right, so our one strand DNA, to make up a strand of DNA, we need to make nucleotides. So I'm going to draw my nucleotide like so. I have a phosphate, a sugar, and then a nitrogenous base. I'm going to make my nitrogenous base look like this, okay? And I'm going to make this nitrogenous base, I'm going to put a C on it. That C means it's going to be cytosine. I'm going to have to add more nucleotides to make one strand. So I'm going to make another nucleotide. And as you can see, the sugar is attached to basically the phosphate of the bottom nucleotide. And sugars look like a little house in this particular case because they're made out of 5 carbons. And if you remember, each corner is a carbon. So 1, 2, 3, 4, 5. That's why we have, it looks like a little house, right? So we have a new nucleotide. Now I'm going to make it look like so. Okay, and that's going to be adenine. I'm going to write A, adenine. Then I'm going to do another one. I'm going to draw two more nucleotides. That one's going to be guanine. And then I'm going to draw the final nucleotide, which is going to be, in this case right here, thymine. Okay, so basically I have 4 nucleotides here. One of my strands of my DNA, I haven't drawn the second strand yet, but one of the strands of DNA is made out of 4 nucleotides. I just randomly made these 4. I could make it longer, right? In fact, in a human being, in one of our cells, we have 3 billion nucleotides in one of our cells. Okay, so it's crazy amounts of nucleotides that make up our DNA. I decided to make 4, and you can see that we have, this is 1 nucleotide, right? This is another nucleotide, another, and another. So opposite to that, we have the other strand. And remember, these two strands interact. And the way that it's drawn out actually is a little bit different. So here I put the phosphates on top. Now on this other strand, in order for it to fit properly with this, the first strand, the phosphates have to be at the bottom. So bear with me, it's going to be a little bit messier, but it basically looks like so. Okay, so that's going to be one nucleotide. You see, one nucleotide, I drew the phosphate, the sugar, and the nitrogenous base. And this one is A. I don't know if you guys can notice at all, and this is going to be like so. I don't know if you guys have noticed at all, but I am taking a lot of time, painstaking time, to make sure that my nitrogenous bases of each nucleotide is fitting like a puzzle piece with the opposite strand. Right? Remember how I drew cytosine like so? And it's the same in this one right here. And that's because it binds to specific nucleotides. And we'll talk about that in just a second. But right now I'm going to finish I'm drawing my third nucleotide right now. My third nucleotide. And then I'm going to be drawing my last and fourth nucleotide. Oops, I went a little bit, this is a little bit wonky. Oh well, it's okay. Okay, so these dotted lines are just the bonds. They're known as phosphodiester bonds. So the actual bonds that hold the nucleotides in one strand are known as phosphodiester bonds. And then in between the two strands, in order for the two strands to stay connected, because at this point they're still not connected, in order for the two strands to stay connected, we have what we call hydrogen bonds. So we form hydrogen bonds between the nitrogenous bases, like this. And this is how the two strands stay connected, okay? So as you can see, if we think about our DNA like a ladder, okay, so I'm going to draw a ladder here. This is my ladder. The sides of the ladder are basically phosphate, sugar, phosphate. I'm going to write an S here just to show sugar. Phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. Same thing on the other side. We have sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. So we say that the backbone of the DNA is a sugar phosphate backbone. Okay, so they have a sugar phosphate backbone. And the actual the nitrogenous bases of the DNA are facing the middle. Sugar phosphate backbone. And then in the middle, okay, in the middle, we have nitrogenous bases, or if you want, you can just call them bases. Okay, and these bases are going to pair with each other so that we have a base pair. So the rungs of the ladder are bases. These are the bases, and they're paired up together because, as you can see, this is how they interact, and this is how the two strands of DNA actually stay together. Now, this interaction between the base pairs, I told you guys I was taking a lot of time to actually draw them out so that they fit together like pieces of the puzzle. As you can see, cytosine is not going to fit properly with thymine. And this is actually what happens in the DNA molecule. Cytosine is only ever going to pair up with guanine, and that's it. So cytosine pairs up with guanine, okay? Adenine is only ever going to pair up with thymine. So if I have a thymine, what's it going to pair up with? If adenine can only pair up with thymine, thymine can only pair up with adenine. So thymine's only ever going to pair up with adenine. If I have a guanine, it can only pair up with cytosine. So this is really important because this is how the DNA knows how to replicate itself, because of this base pairing rule. Okay, this base pairing rule, which you are going to need to know. Okay, so in the exam, if I give you an exam question where I tell you one strand of DNA, I tell you that the strand of DNA looks like so, right? From this one strand of DNA, you can tell me what the second strand looks like. How? Because of this base pairing rule. You're going to tell me that adenine binds to thymine. Cytosine binds to guanine. Ooh, we have another cytosine here. It binds to guanine. Guanine binds to cytosine. Thymine binds to adenine. Adenine binds to thymine, and thymine binds to another adenine. And this is what our double-stranded DNA looks like. So in the exam, I may tell you just one strand, and you have to tell me what the second strand is of DNA, right? So this is two strands that make up one DNA molecule, one DNA molecule. Another thing that I want to bring to your attention is, as I mentioned before, the phosphates on this side are facing up and the phosphates on that side are facing down. This is because DNA and the two strands of DNA are what we call anti-parallel. Okay, anti-parallel. They go in opposite directions. It's not a one-way street. If it was a one-way street, there would be no way that you can go back to the beginning, right? If you leave from your house and you have to go to, say, Walmart, yes, you go down the one-way street, but then you can't go back to your house if there's no other street, right? If it's a one-way street, you can't go back. With DNA, we have one-way street, and then the other street goes in the opposite direction, so that you can go from your house to Walmart, and then from Walmart to your house back. So it's a two-way street, and this is known as being anti-parallel, okay? And so we call this side, it goes from 5' to 3', and this one is going to go opposite. It's going to go from 5' to 3'. So if we go back here, this can also be 5' to 3', like so, right? And then the opposite strand, the strand that is complementary to the strand because it pairs with the correctly with the bases. So it means that it's complementary. So base pairing is also the word complementary. Okay, complementary just means adenine with thymine, thymine with adenine, cytosine with guanine, guanine with cytosine. The complementary strand, it goes in the opposite direction, so it's going to be 3' to 5', and that is known as anti-parallelity. It's really important to be anti-parallel in DNA because if this was going in the same direction, these would never actually fit. Like molecularly, with all the atoms involved, this would not actually be able to fit. And so with that, we have our DNA, right? As you can see, we have this is only one strand of DNA, but there's two strands. And so you can see in this picture right here, there's two strands of DNA, each made out of nucleotides, and they come together with specifically, okay? Here you just have the actual molecular structure of what adenine, what the nitrogenous bases look like. I do not need you to remember them. I just need you to remember the names of the nitrogenous bases. I wanted to put this out there to show you guys what the actual difference between them are. Okay, so DNA is a double helix that stays together, and the strands, as you can see, are made out of nucleotides. And the nucleotides base pair specifically with complementary rules, so adenine to thymine and thymine to adenine, cytosine to guanine, and vice versa, with hydrogen bonds. So the hydrogen bonds are holding the actual nitrogenous bases together, and thereby they're holding the two strands of DNA together. And as you can see at the back, at the actual sides of the ladder, you have the nitrogenous bases are found in the middle. On the sides, you have just the sugar and phosphate parts of the nucleotides. So we call it the sugar phosphate backbone. This is where we talk about anti-parallelity of the strand. So you see how it's one's one way and the other one's the upside down, and that's known as being anti-parallel. All right, so that was DNA, and remember DNA works for it actually bears the information that codes. This basically bears the information, or it holds that code. And now we're going to talk about RNA, which is what helps us make the proteins from our DNA. So RNA is also a nucleic acid. That means it's also made out of nucleotides. But there are a few differences between the structure of RNA and DNA, and this is critical because if they look the same, they would do the same function. And obviously they do not have the same role, and therefore they must look different. So the nucleotides of RNA are very similar to the nucleotides of DNA. They're also made out of a phosphate, a sugar, and a nitrogenous base. But the sugar is actually known as a ribose, hence the name ribonucleic acid. Okay, so RNA is called ribonucleic acid because the ribo, right, is in reference to the ribose sugar. It also has a phosphate group that doesn't change. And then it has nitrogenous bases as well. The nitrogenous bases, some of them are the same as DNA and then others aren't. So it has adenine, it has guanine, it has cytosine, but instead of thymine, it actually has another one known as uracil. Okay, so uracil takes the place of thymine. So RNA does not have thymine. So if you guys can look at what I'm writing on my PowerPoint right now. If I write something like that looks like this, AGGUAC, okay, because it has a U in it, which stands for uracil, I know that this molecule right here is actually RNA. If I wrote AGGTAC, T stands for thymine, and that means that this molecule is DNA, okay? So the same complementary rules apply to RNA, where adenine pairs with uracil, right? Uracil, not because it doesn't have thymine, and uracil binds with adenine, guanine binds with cytosine. The other thing about RNA is that it is single-stranded, right? So it doesn't pair up with itself, but like with another strand. But it's important that we have these bases are able to base pair with each other, and they're complementary to each other, because this is how we read, how RNA reads DNA, okay? This is how it reads the DNA. So for example, in my exam, I may ask you guys, so stop share. I may give you a strand of DNA. So I may give you a strand of DNA. It'd be T, C, G, G, C, T, A, T, A. Okay, so that's my strand of DNA. I know it's DNA because it has thymine. Okay, I could ask you, from this strand of DNA, give me its complementary strand of RNA. Okay, so give me its complementary strand of RNA. And so you would look to see, what the nucleotides that a base pairs to. It's obviously going to be different nucleotides because it's RNA now, right, and not DNA. And so T is going to bind to A because RNA also has adenine. C is going to bind to guanine because RNA also has guanine. G is going to bind to C, cytosine. This other G is going to bind to C. C is going to bind to G. T is going to bind to A. And then A is going to bind to U, right? Normally in DNA, A would bind to thymine, but we do not have thymine in RNA. So adenine binds to uracil. T is going to bind to TA, and A is going to bind to U. And then afterwards, as we'll see later on, these two actually separate, right? And the RNA goes on its own way to continue with protein synthesis. And then from this, we can make our actual protein. Okay, so I may ask you to either make the second strand of DNA or go from a strand of DNA to a strand of RNA. Or I may ask you to tell me, okay, if I have AUG, is that DNA or RNA? And you'll say it's RNA because it has a uracil in it. And if I have ATG, ATGA, let's say, I don't know, the number of nucleotides doesn't matter, you'll say like that is DNA because we have thymine in it. So you definitely need to know the differences between RNA and DNA, not only in their role, so know that the roles that RNA takes and the roles that DNA has, but also know that RNA is made, the sugar in its nucleotides is ribose instead of deoxyribose, which is what we have in DNA. RNA is made out of one strand instead of two strands, which is what DNA has. DNA has two strands. And then in addition, the third and final difference is that RNA, its nucleotides are adenine, guanine, cytosine, and uracil. So it has uracil instead of thymine. Okay, and you can see that it's just one strand right here. So DNA has two strands, RNA is one strand, and then it has uracil, etc. So definitely watch these two videos on my PowerPoints. This is also another video on my PowerPoint. And that's where I'll end the actual lecture today. And I'll see you guys in the next lecture to continue chapter 10. But go over these particular videos, definitely do so. See you guys later. Bye, guys.